Right, boys and girls, we have got meiosis today. Meiosis is the cell division in the reproductive organs or in sexual reproduction. So, keyword here is gametes. Gametes are sex cells, or also known as the sperm cell in males and the egg cells in females. Now, gametes are produced in the reproductive organs, such as the that is not good. Such as the testes and the ovaries. Gametes have 23 chromosomes each, so a sperm cell will have 23 chromosomes, an egg cell will have 23 chromosomes. This is also known as haploid, a haploid number, so that's 23, and diploid, just remember D as double, so that's 46 chromosomes. Now, fertilization. Fertilization is the fusing of a sperm cell and an egg cell, and that makes a zygote. Not really too much about zygotes, just makes a zygote. Sperm cell plus egg cell equals zygote. So, fertilization, you need to know, is a random process. So any sperm cell can fuse with any egg cell, and no sperm cell, no egg cell will be exactly identical. They all have a little bit of change, so they're all alleles. But basically, because it's a random process, it is there is genetic diversity, and that is very key, because genetic diversity means that things can't die out easily, isn't it? Because mutations and all that, if you have genetic diversity, things will survive, because not one thing will kill out everything, basically. So know that randomness creates genetic diversity, and I can almost guarantee you that will come up in an exam somewhere. So that is very, very key to know that. Right there. So, I'm not going to talk too much about PMAT1 and PMAT2 at this very minute in time, because you should already know PMAT1 is in mitosis. You've got prophase, metaphase, anaphase, telophase or telophase. But I will talk about interphase. Interphase is just before meiosis, so it's like the preparation for meiosis. So in interphase, the chromosomes duplicate and they unravel. So it's almost like they unravel into like loads of long strands, basically, like that. That's really it for interphase. They duplicate and they unravel, so you'll get 4 lots of chromosomes. So you'll start out as 2 because it's a normal cell and it's diploid, and then it will duplicate and it will become 4. So this bit here is prophase. So I'll write the prophase in here. I'll just write pro here. So this is the prophase, this bit. In prophase, chromosomes condense, and they also pair up with a homologous pair, or homologous. That's how people say it, homologous. I can't even say it. Homologous. There you go. Homologous pair. So a homologous pair is, I've got that on another page actually, I think, but I will tell you now. It's the same size. So a chromosome, 2 chromosomes which are pairs in the homologous pairs, have they're the same size. They've got genes in the same spot, and there is something else which I cannot remember, but I will show you that in a sec. So yeah, something special happens. So we need to know that it condenses, homologous pairs come together, and there's this special process called crossing over. Now, crossing over is like when the homologous pairs, they come along and they almost like interlock with each other. Now when they interlock, chromosomes exchange their genes. So genes is a part of the chromosome, and they will exchange genes. So this is where I've got the black chromosome and the brown chromosome, and they're homologous pairs, so they are literally almost identical but not fully. Same size, same genes in the same spots, and they will swap. So parts of the black gene will go into the brown gene, and part of the brown gene will go into the black gene, like this. So you've got genetic diversity again. Moving on, we will go on to metaphase. So metaphase 1. So that was prophase 1, and this is metaphase 1. So we've got the homologous pairs line up along the middle of the spindle. So in mitosis, we know that there's a spindles formed, like spindle fibres made out of microtubules, and the centrioles like this, forming the spindles and all that. But don't need to know too much about that. But just know that metaphase, take the M and it makes middle. So M, middle, homologous pairs line up along the middle of the spindle or the equator. That's the correct word to put, equator. So moving on to anaphase. Anaphase, take the A and put apart. So homologous pairs separate, move to opposite ends of the spindle, and you almost get this sort of separation of the two homologous pairs that way. Now, because they've duplicated at the start in interphase, and then they condense, they move apart, no, they go to the middle, and then the anaphase, they move apart to opposite ends of the spindle like this. But we need to know that homologous pairs, it's a random decision made by whatever to either, so these are the two homologous pairs, one here and one here. Now these two can go this way and these two can go this way, or these two can go this way and these two can go this way. So I've got the two diagrams here which explain the two different ways of going random order, and this also creates genetic diversity because it is called independent segregation of chromosomes. It means they can move in different combinations. Yeah, so that creates also genetic diversity. So genetic diversity is a very key word as it comes up a lot. Oh no, I just ripped off. No, I haven't. Good. No, decent. Now we're on to telophase or telophase 1. So now we know that the chromosomes have moved apart in anaphase from the A apart to opposite ends of the spindle. And basically the cell divides into two because we've got these two ends of the spindle, the two chromosomes here. Now basically cytoplasm closes, the cell binds around and it literally breaks off, and this is called cytokinesis. So that's really meiosis stage 1. So now we're on to PMAT2. And PMAT2 consists of prophase 2, which is the cell makes spindle and other processes happen. So there's not really too much on it because in prophase 1 there's loads of stuff going on about condensing and crossing over, so really interesting stuff. But in prophase 2 there's nothing really interesting to know. Just the cell makes all the spindle fibres and all of that. Then a metaphase is literally the same thing as meiosis stage 1 or meiosis 1. Things move to the middle of the spindle. So these chromosomes move to the centre of the spindle, or the equator of the spindle. Now anaphase. So in anaphase 1, we've got the chromosomes from the middle sort of like separating to this different ends in there. The homologous pairs separate, so that goes that way, that goes that way, and that goes that way, that goes that way, or the other way around. Well, in this one, you've got half of the chromosome, so the chromatid going one way, and the other half of the chromatid, the other half of the chromosome, which is the chromatid, going the other way. So you've got this separation of the chromosome as the centromere splits and the centrioles and the spindle fibers contract, which pull the halves to the other ends of the centrioles. Yeah, so that's anaphase. Just know that it's apart, like I've said before. Take the A apart, centromere splits, chromatids move to opposite ends of the spindle. Now the final stage is telophase. Telophase is the cell division. So this cell divides into two again by cytokinesis. So you end up with 4 daughter cells, because in the first stage you've got your one cell, and then you've got the 2 cells which are here, and then these divide into 2 again, and you get 4 separate cells. Yeah, so then that's all meiosis. Now I'm just going to talk to you about the differences between mitosis and meiosis. So this may be quite a common question in an exam. So I'll talk about mitosis, and then obviously you can take the opposites. I'll say the opposites for meiosis. So in mitosis, 2 daughter cells are created. So as we just learned in meiosis, 4 daughter cells are created. Now in mitosis, genetically identical, all the cells are genetically identical each other and to their parent cell. Well, in meiosis, they're all different. Every single one is different, so this is where it comes into the genetic variation or genetic diversity. So every single one is different, and it's different to its parent cell. Now, in mitosis, it produces cells with full number of chromosomes. So this will be a diploid number, but as we know in meiosis, it creates 4 daughter cells where they each have 23 chromosomes, because we saw in the last stage of anaphase, I will try and show you, in the last stage of anaphase 2, we've got these 2 chromosomes which have the full amount, so that's, what's that? Oh, I'm getting really confused now. They're 46, that's a pair, so when they split, it makes 23 and 23, and that divides. So, yeah, produces cells with full number of chromosomes, and that is it. Well, mitosis, the cells repair. Mitosis is used for like cell repairment and all of that. Meiosis is used for sexual reproduction. So there's your 4th one as well. Thank you for watching. That is meiosis. Like and subscribe. I will catch you in a bit.